Good morning, everyone. It's a pleasure for me to introduce Professor Rohde. Professor Rohde is assistant professor at the Burnett School of Biomedical Science, University of Central Florida in United States. His research focuses on the understanding of tuberculosis pathogenesis and by several approaches to address its control, such as regulatory pathways involved in mycobacterial tuberculosis, intracellular survival, and stress response relevant during human infections. Further, Dr. Kyle, development of new diagnosis methods, especially point of care platform, and also discovery of new drugs for the treatment of TB, included from marine natural products. It's important to highlight that currently Priscilla Halick, that get a PhD by Health Science Program of PrInt-FURG, is postdoc of Kyle team. I would like to thank Dr. Kyle, Professor Rohde, to participate in this important workshop for our institution. Go ahead.
All right.
Thank you.
Let me try to share my screen here. And can you everybody see the title page okay?
Yes.
All right. Well, it's an honor to participate in this workshop focused on internationalization and CAPES related things. I've had a fairly long connection with your region of Brazil and with the CAPES program. My first PhD student was from the University of Pelotas, I believe. And that was how I became introduced to Pedro quite a few years ago. I had an undergraduate in my lab a few years back, also supported by the CAPES fellowship. As well as Priscilla before the pandemic, was sent home early, but thankfully we were able to get her back here after her PhD was completed. And hopefully one day post-COVID we can have others that were scheduled to come. I know Ana Julia we had other students that were supposed to come and exchange, but those plans were changed, but that's all right. We're doing the best we can here. So as Pedro said, my stuff I'll talk about today is sort of two unrelated stories, but both sort of nature-inspired approaches to diagnostics and TB drug discovery. Let's see if my computer will work. There we go. So this is my lab at the University of Central Florida. Just to give you a little perspective here, in case you are coming with your children to visit Mickey over here, we're very close. So you can arrive at the airport and then come by my lab and have a visit and go to Disney and call it a business trip, if that will help with your taxes or something in Brazil. So we're blessed to have a beautiful campus that right now is very small. The big campus is about 30 minutes away where I teach, but we're at a new campus where there was space to build the medical school and the research part. So this is where the research is happening. And Pedro already kind of gave you the overview. My interests are a little bit diverse from my background of gene regulation and genetics, and now we've moved into some more translational areas as well. So the couple of stories I'll share today come from sort of the drug discovery and diagnostic areas. So many of you may be aware of this sort of background information of significance, but our lab focuses mostly on TB and related mycobacteria. And I had to update my slide because we used to be able to claim that it was the top infectious disease killer, but hopefully again, maybe by next year we can change back the slide, but it's still a very important infectious disease that kills as well as affects millions of people each year. And the main reasons why it's still such a big problem is basically we need better of everything. We need better drugs and diagnostics as well as vaccines. But because I don't know that much about vaccines, I won't talk about that element of need today. I'll try to focus on these two that I maybe know a little bit more about. So as I think of sort of the challenges that we're facing trying to to treat TB better than we currently can. There's a number of challenges that we keep in mind. The most obvious is the number of drug-resistant strains that emerge. So you have about 500,000 cases each year that are resistant to multiple frontline drugs, and there are numerous designations of extremely drug-resistant or totally drug-resistant that get worse and worse. So one of our goals is to find completely novel drugs that hit different targets that may work on the strains that are already resistant to the drugs that we have. Another real challenge that's tied to sort of the biology of tuberculosis is the issue of phenotypic drug tolerance, where the bacteria in the lungs, in the host, are surviving inside a relatively stressful environment inside a macrophage. And those macrophages are inside lesions, the granuloma, that have low oxygen, they have some nutrient limitations, acidic pH, all sorts of stress that the bacteria can survive, but they stop replicating. And so we have these dormant persister bacteria that aren't killable with existing drugs that target the cell wall or the ribosome or something you only need if you're trying to grow. So that's another one of our focus areas, is how do we change the way we go about drug discovery to bias our discovery to things that maybe can kill these hard to kill forms of TB. And a third sort of more pharmacological challenge that is very real in TB is the difficulty of getting the drugs to where the bacteria reside during the infection. And so it's hard enough to have a drug that is safe, that is orally bioavailable, that can even get to the bloodstream. But with TB, it also has to accumulate in the lungs and get into the macrophage and get inside the granuloma to reach the bacteria. And studies starting maybe 10 years ago by Dr. Veronique Dartois' group, they were able to show with some very cool mass spectrometry imaging to measure where in a real TB lesion do the drugs accumulate. And we imagine if you give somebody 4 drugs, that all 4 drugs go and attack each bacteria so you can kill them quickly and try to avoid resistance emergence. But what those studies shown is that that's not the reality, is that some of the drugs will get stuck at the periphery of the lesion, some go inside, but they stay outside the cells. One of the drugs accumulates nicely in macrophages. So you really don't have equal treatment with 4 drugs, depending on the environment of each bacteria. Maybe they're only feeling 1 or 2 of the drugs. So this is a challenge that we face. I'm not going to talk about how to solve that problem. I will leave that one to smarter people than me. One other interesting aspect of TB that has come about in the last few years is this concept of sort of the heterogeneity of the bacteria in an infection. That we usually study them in vitro where they're growing in nice, happy media. They're all sort of thought to be the same. But now we know from single cell studies that even in vitro, bacteria are growing at different rates. They're more or less susceptible to drugs. Even in one macrophage, one bacteria on this side can have different gene expression and different drug susceptibility than a bacteria in the same cell. So how do you incorporate this complexity into our drug design? So basically, we have to hit more than one target with the drugs that we're looking for. And one other study that I thought has always stuck out to me from 2016, I believe, was a study in human patients using this FDG labeling and PET CT scanning that showed that even within a patient, some granulomas respond to treatment, while other granulomas may continue to grow or not be cleared at all. And the scariest result to me of this study was that there were patients that after 6-month standard treatment and a 1 year of follow-up, they were declared clinically cured, but these FDG PET imaging showed that they still had active lesions in the lung, even though they weren't shedding bacteria, they didn't test positive, and there was TB RNA found in these lesions. So now even people that we declare cured may still have, quiet hiding out TB hanging out, perhaps to reemerge later. So these are all the kind of things that make this a challenge. And So I'm going to tell you about some of, there's many scientists and many groups much bigger than mine, but we're all trying to think of innovative ways to find new drugs. Because if you keep screening the same drugs on the same in vitro broth culture bacteria, we're not going to find anything new to solve these problems. So I'm going to tell you a little bit about a model of dormancy that we try to mimic this stressful, non-replicating kind of existence of TB in vivo and apply that in drug discovery with the hope of finding drugs that can take out these tolerant bacteria. And although we have a lot of different drug projects going on that explore these different strategies, today I'm going to mainly focus on two of our sort of marine associated natural products type stories. So before I get too far into it, just briefly tell you the main sort of tools or assays that we've been using. The first for the drug discovery for sort of a standard model of replicating TB, how we've always done it. Because TB grows so slow, we don't like to wait for colonies to grow because that's 1 month for every experiment. We use reporter strains that are either fluorescent. So this is a colony of TB expressing M. cherry, it's a very beautiful colony. Now we've switched to using mostly luminescent TB expressing luciferase. So they're autoluminescent, they glow very bright, much better sensitivity. So we are blessed to have a robotic liquid handler that my students and Priscilla now are very happy to have instead of the hand manual pipetting to dispense lots of drugs into 384-well plates. Then we can add the glow-in-the-dark TB and wait a few days, and the ones that are dead will stop glowing, and the ones that are unaffected will give a high signal. So it's a fairly straightforward assay to do MIC curves or to study antibiotics. But a different assay, a modification of this that we have relied on quite a bit is what we call a multi-stress dormancy model. And this was actually developed by Dr. Kalata, who was the director at my department that gave me my first and only faculty job so far. So I'll always be very thankful that he took the chance on me. But what they did was basically develop this combination of stresses that we believe are relevant to sort of in vivo life of TB. So if we grow TB with sort of mild nutrient starvation, low pH, low oxygen, for about 9 or 10 days is sort of optimum, the bacteria adopt most of the phenotypes that we attribute to dormant bacteria that you find in patients. So within 1 cycle, they halt their replication. They did some staining studies to show that their cell wall changes and they lose their acid-fast staining. So that's sort of the green line. They accumulate lipid droplets, which is how we've seen in bacteria coming out in patient sputum that they're enriched for bacteria full of lipid droplets. So in this model, the bacteria also do that. They did some transcriptome studies and said, okay, all the genes that we expect to be going up when the bug is hypoxic or starving, they have the normal profile. But most importantly, after about a week in these conditions, the bacteria become very resistant to the frontline drugs. So we adapted our luminescent TB model to discover drugs that could kill these hard to kill bacteria. And I won't tell you the 1.5-year story that it took to get to what seems like a very simple model. You can ask Carolina, my former PhD student who is now back in Brazil, about that little adventure in science. But we now have an assay that we like in 2 days treatment. So just to convince you that in this model, the bacteria become very resistant, this is what a dose response curve looks like with rifampicin and isoniazid. That if you kill replicating bacteria in oxygen and rich media, you can see that at very low concentrations they are killed. But the dormant bacteria, you see a dramatic shift to the right of close to 100 or more fold increase in resistance to these frontline drugs. So we thought, well, if we can find any drugs that can kill these bacteria, even remotely close to the replicating bacteria, this would be very novel type of compounds that may give us a way to kill dormant bacteria better than our frontline drugs. So we first implemented this assay and both assays in a collaboration with Dr. Amy Wright at the Harbor Branch Oceanographic Institute, a couple of hours from here at FAU. And thankfully, we convinced NIH this was worth doing, so we were able to work on this for several years. So what Amy's lab had done over quite some time is to develop a library of marine-derived natural products that they had a remote-controlled robotic submarine that they could, like a video game, drive down to the bottom of the ocean and pick up these sponges and exotic ocean floor organisms. And then they would, instead of a crude extract, they would do a single stage of purification so that they're semi-purified samples. So they had a library of about 4,500 what they called peak fractions. And we then screened these samples against replicating and dormant TB. And then with help from Amy's lab, we sort of did the classical activity-guided purification to find what is the component that is doing the killing. So some of these mixtures had only one component, or 90% pure. Some maybe have 10 different chemicals in there, but they're much simpler and easier to move forward than a complete mixture of a sponge that was extracted. So this is an overview of the screening of this entire library that just shows sort of the percent killing against the replicating TB or the dormant TB. The gray ones are ones that we sort of eliminated from future study at the time because they were cytotoxic to mammalian cells, which we wanted to avoid. And so we kind of narrowed our list to the black dots, and most of them would kill the replicating TB, which are easier to kill. But you'll notice there are quite a few samples sort of above the line vertically that were able to kill dormant TB in our model. And some showed dual activity. So the ones with arrows are just ones that I will sort of show you some of the compounds that came from those. But we have some other work on some of these others. But it takes a while when you have to purify and back and forth for months with the chemists to find the active component. So these are just some example sort of activity profiles of the kinds of things that we discovered. Some we found that were the black lines are replicating TB. So this was a molecule that, like many drugs, kills active TB, but didn't kill dormant TB. But the ones we were most interested in were the ones that showed comparable activity in both models. So these are a few examples. And then what became our favorite was this guy that could kill dormant TB almost 10 times better than replicating, which is, in all the screening we've done, is a highly unusual profile to kill better. Usually it's 10 times worse or 20 times worse or more. So we were really interested in this compound. So these are some of the structures that the lawyers at the university say we're allowed to show in public at this point. This crazy molecule here is the one that doesn't kill dormant TB. We had a few other examples that were dual active, but the ones that we have spent more time studying were these two related compounds that came from the same sample, the same sponge, and they belong to a class of compounds referred to as pupihenones, or it's a Hawaiian word that I don't know how to pronounce, and it was named after some Hawaiian princess. But nonetheless, we found these two compounds that number 4 is the one that has this profile. So this was exciting, but we wanted to do further studies, and you can only have so much sponge from the ocean in the freezer of the collaborator. So eventually, if we want to pursue this, as with most natural products, you need to find a synthetic route to make this compound and to develop it chemically. So we had in mind to understand the mechanism of action of this drug and also to improve the properties. So thankfully, I met at a conference a chemist who was willing to take on this challenge. He does whatever chemists do and says, I think I can make this from things we can buy commercially. And so we were able to get some funding to continue this project. And so Dr. Suchek at University of Toledo successfully made the core structure and about 29 or 30 synthetic analogs where we tried to make variations at different positions to understand what part of this molecule is important and how can we make it more active than it was originally. So 15 of these analogs were not active. So I'll show you kind of what we learned not to do with the compound, but about half of them maintained activity and showed some interesting profiles. So this is just a brief summary of the activity, the MIC, against replicating and dormant TB. The ones at the top were ones that looked kind of like the parent compound, that they were 4 or 5 times, sometimes 10 times more active against dormant than replicating TB. So that made us confident that this profile was not just an accident, that we can modify, and every time we could see this enhanced killing of dormant TB. Some of them we make modifications and we lost some activity. The most interesting to me were these two in yellow, that the modifications improved the activity against replicating TB while keeping the activity against dormant. So we're very interested in kind of understanding what is the mechanism behind these profile. So this was sort of my attempt to summarize what did we learn about this compound. You know, we learned that this part of the ring structure, you have to have a closed ring. If that ring is open, it has 0 activity. Our 2 initial compounds, one of them had a methoxy group here, but we know we can remove that and the activity is maintained. But if you attach a very large group here, you lose activity. We learned some of the same kind of things if you modify the top end. Some modifications improve the activity and lead to these yellow compounds. Some modifications completely obliterate activity. So the stage we are now is we've sort of scaled up the synthesis of several of the most favorite molecules. So we now have enough to begin sort of the mechanistic studies. So Priscilla is working sort of optimizing our selection of spontaneous resistant mutants in TB, which is a slow process. So we hope to find mutants that can resist this compound, where we have a colleague here who can do the whole genome sequencing, fairly straightforward, and hopefully to identify which gene can be mutated to achieve resistance and maybe lead us to the target. We're also, very soon we'll be doing a transcriptome profile. We've had some about 1.5-year complications trying to get our RNA-seq back to working, and COVID did not really help that at all. And Dr. Suchek has also been making modified drugs that have an affinity tag that will allow us to react with the target proteins and hopefully to pull down or precipitate potential targets. So these are many sort of standard ways that we're trying to use to understand how do these unique molecules kill dormant TB. So we're fairly confident it's going to be a new target, and this is a new type of structure, but we have some work left to do to improve the potency and understand how it works. So kind of a second story in our adventures in marine-related natural products. We're in collaboration with Dr. Bill Baker from University of South Florida, about 2 hours from here. And this is a picture of him in a t-shirt in Antarctica, where he goes several times a year as part of research teams to collect exotic fungi and samples from extreme environments to see what kind of natural products they may be producing. And so the interesting aspect of the approach we've been taking with Dr. Baker comes from the idea of, you know, when you take fungi from the soil or from the Antarctica, wherever, and try to grow them in artificial media, there are many of their metabolic processes that are completely turned off when they are not being triggered by competitors and environmental stresses. And so if you screen natural products in the normal way of growing in whatever the artificial media is, the idea is we're missing a lot of potential natural products with, you know, interesting bioactivity. And so what they have taken to doing is treating their fungal cultures with compounds that will affect the chromatin structure. So either histone deacetylase inhibitors or DNA methyltransferase inhibitors that can open up regions of chromatin that were otherwise silent and activate these gene expression of additional metabolites. So this was sort of a pilot study that we wanted to say, could we discover TB active compounds that we would have missed if we did this the traditional way? So what Bill's lab did is they had about 500 different fungal samples, and they either grow them the normal way and make an extract, or they treated them with one of those classes of inhibitors and then made the extract. They sent those to us and we screened them against TB. So not too surprisingly, we found some samples that have activity, but it was not affected by these, what we call the epigenetic modulator treatment. So we have very strong inhibitors. So we're interested in these and we're trying to purify these components. But the most interesting samples were ones that look like this, where the fungus grown in the normal way, we would have had little to no activity. We normally have a cutoff of about 50% activity to be even interested. So these we would have all said, these are not a hit. But you can see from the ones with a spike in the blue bar that if you treated with a methyltransferase inhibitor, that somehow activated the production of a metabolite that was very good at killing TB. And so we are working back and forth with Dr. Baker's lab now. We have a few of these that are, close to pure, so we can try to determine structures and move forward from there. But this was sort of an exciting new adventure for me in natural products, is sort of harnessing the power of epigenetics to awaken natural products that otherwise would go undiscovered. So if we can now convince the NIH that this is an exciting of a project as we think it is, to have a little funding, then we can go a little further, a little faster. But that's probably the struggle of scientists everywhere. You know the stuff you're working on is cool, but the people with the money don't think it is as cool as you do. So before I kind of transition and talk a little bit about some of the diagnostic stuff, just wanted to acknowledge those that were involved in these projects as well as others I don't have time to talk about. So Carolina was my first PhD student that started the marine natural product project that led to discovering that pupihinone compound. The others shown here, Brevin and Rashmi, are still with me, but they're working on different projects, and the rest have moved on to bigger and better things. So at the moment, Sandy and Priscilla are the ones carrying the load of the many 10 or 12 drug projects we have going, because if somebody has something in their freezer and they want to know if it kills TB, I almost always say yes. So they will send it to us and maybe we have a project. So enjoy collaborating where we can. So all of these projects are at the stage now of trying to understand the mechanism of action of these natural products, as well as how can we synthetically produce them with the help of a chemist to move them forward as TB treatments. And so the hope is that using this dormancy model will help, prioritize finding of drugs that can kill dormant TB. So I'm going to shift gears sort of the second half and talk about this diagnostic project that I had no experience in diagnostics when I came to UCF as faculty. My background was in TB and macrophages and things like that. But I was fortunate to meet the brains behind the platform. And then we've just tried to merge our skills together to apply this to tuberculosis. So the current team is with Dr. Yulia Gerasimova, is the chemist right now behind the platform, as well as Dr. Kopashnikov, her husband, that are in chemistry. Dr. Brian Kim, I'll talk at the end about his work. He's a biomedical engineer that is helping us make the 3D printed device that we will use for this diagnostic. This was me sort of in the post-COVID mode. And we've been fortunate now to be able to partner with FURG and Pedro and Andrea and their group down there on an NIH-funded project where we hope to sort of optimize the platform in Florida and then send it to Brazil and test it more in the real world and to work along those sides. So there's a lot of students and technicians that have been involved as well. So why do we need more TB diagnostics? There's many options out there. Chest X-rays you can do and detect if you have lung damage, and but sort of early detection maybe not the best method. Smear microscopy is probably the most widely used at in most endemic areas because it's cheap and fairly easy, but not so good as far as sensitivity. And you also aren't able to distinguish tuberculosis from related bacteria that require completely different treatment regimens. Culture is the best, but it's very slow and has, biohazardous considerations as well. And now there are many DNA nucleic acid based tests, such as the Gene Expert, which are very good, but also have some limitations as far as implementation in resource poor settings. I also grew up in South America, not too far from Brazil, and the electricity would go out. So if you need to have your instrument plugged in, that can be a problem in many places that can't keep the lights on very well either. So the platform that we are working on is definitely at a very early stage, but we think has some very good potential. And so it is inspired by the naturally occurring ribozymes, which are RNA molecules that can have catalytic activity. So that was kind of the inspiration of this idea. And so if the nucleic acid folds in a certain conformation, this class of ribozyme can cut DNA or can cut other nucleic acids. So for the diagnostic, our collaborators have taken this RNA idea that nature uses and converted it to a DNA-based platform, where now we take the two, so they found where you can split the catalytic core in half. So you can see we call it DZA and DZB for deoxyribozyme. So now that's the binary part of the name. If we attach these arms to it, these are just strands of DNA. Then nothing more, just oligonucleotides that you can buy from a company. We can design arms on there that will recognize TB DNA. And so if we move down here, if TB DNA is present, the two halves of this sensor will hybridize right next to each other. And now you reform the catalytic part of the deoxyribozyme. On the other side, we have what we call the reporter binding arms. So they will bind to a fluorescent substrate or reporter that is quenched. But when this assembly occurs, now the enzyme part will cleave this little strand in half, the quencher floats away, and now you have a fluorescent signal. So it's a very inexpensive, if you want a new sensor to detect a new DNA, you just spend about $10 or $15 and redesign this portion of the sensor. They're very sensitive to detecting single nucleotide polymorphisms. So they can also be used to detect drug resistance mutations or TB versus other related species. So with this platform in mind, I just wanted to kind of show 5 or 6 slides kind of illustrating what we think this platform can do and what we're trying to accomplish. So it's sort of an initial validation where we designed a panel of sensors targeting different genes associated with drug resistance in tuberculosis. So we can either design sensors that detect a wild type sequence. So you have a gain of signal. If it's wild type, you get a signal. If it's a mutant, drug resistant, you lose the signal. So as you can see here, all of our sensors correctly can identify wild type for that target sequence. But if we have a drug resistant mutant in that gene, you see no signal. So we can discriminate very well. We can also design sensors that positively detect the mutant version that would be drug resistant. So now you see the opposite profile. We get a signal if it is drug resistant, but if it's wild type, you get no signal. So we think there's an advantage to this detection of mutant over hoping for a loss of signal if it's a mutant, because if you get no signal, what if your assay failed? You don't know for sure if it's a mutant. It may be just a failed assay. So we like this design of detecting the mutants, but one problem is that if wild type is a G and anything but a G means it is drug resistant, you have to detect 3 different possibilities of mutation without knowing which one it is in reality. So we tested the idea, can this assay, what if we mix together 3 different arms, as we call them, that can detect any 3 of the mutant possibilities? And we mix all 3 of these together, could we detect any mutant allele? So this is just a simple graph that shows each sensor. So the sensor to detect a G can detect the G, the sensor for A can detect A individually. Now, if we mix them all together in the same tube, we can detect any mutant, any version of that allele that is not wild type. So it just illustrates the sort of the simplicity and flexibility of the platform to mix together multiple sensors and detect more complicated, target sequences. One other clinical scenario we wanted to imagine is heteroresistance. So often in the clinic, you may start with a drug susceptible strain, but then during the treatment, you may have one clone that mutates to become drug resistant. And now it's starting to grow, but maybe it's a minority. So the patient goes for detection and say, do I have susceptible TB or drug resistant? Well, if it's 90% of your infecting strain is wild type, the gene expert and others are not very good at detecting that tiny population of drug resistant. So we modeled that, and basically we mixed together 90% wild type and only 10%, we even went lower, but 10% mutant. And this graph here just shows that with our sensors, we can detect the mutation, the drug resistant minority population, even when it's as small as 10% of your infection culture. So this we thought was a sort of an advantage potentially of this platform compared to things like GeneXpert that look for the loss of signal. So you can't see that if 90% of your sample is wild type. So that's another, we thought, an benefit of this type of assay. And as we tried to imagine with lots of input from Pedro and others thinking about the real world of what does this assay really need to do, we thought, well, in many cases, fluorescence and detecting fluorescence requires a more expensive machine to detect it. So could we imagine a version of this platform where we can eliminate fluorescence and have a visual detection? That'd be very simple, very cheap, hopefully. So I'll tell you how we've tried to do that. The other challenge is many assays that we were working on involve PCR. So to amplify the target sequence for better sensitivity, and then we would detect with our sensors. But you need a thermal cycler and electricity to do PCR. So we began to explore methods of amplification that don't need PCR. So isothermal amplification. And then finally, we met Dr. Kim, who is now right upstairs from me, who said he could 3D print a thermal cycler for very cheap that can run on a battery and have Bluetooth to send the data to your phone, all this fancy stuff. So we said, oh, maybe we don't have to eliminate PCR. Maybe we can use PCR as long as we know this cool engineer upstairs that can make a cheap thermal cycler that doesn't require electricity. So I'm going to show you just one slide from each of these attempts to improve on this platform. So this again is not my work of my brain, but of my chemistry collaborators. So to achieve visual detection with a color change, Yulia was aware of another type of deoxyribozyme. So a piece of DNA that could have enzymatic activity was this G quadruplex structure that it will spontaneously fold, can bind hemin, and then it gives peroxidase activity. So you can use substrates like for Western blots that will change color. But the question is, how do you connect that with our sensors for the detection of the TB DNA? So what they did is design this little hairpin structure where this is the G quadruplex. They added this domain to hybridize to it to keep it from folding and becoming the enzymatic form. So it's locked in an inactive form. But if the target DNA is present and our sensors, just like the ones I told you about before, can hybridize, now instead of cleaving a fluorescent substrate, they will cleave this inhibitor domain off. The enzyme part is free, and now it will spontaneously fold and bind hemin and give a color change only in the presence of the specific DNA. So to test that idea, we tried to design an assay where you could tell what species of mycobacteria is present, TB or abscessus or Kansasii. So we identified a hypervariable region of the 16S ribosomal RNA that was different between all these species. And then we found a universal sequences
flanking that. So we have a pair of universal primers that will work on any mycobacterial species. And then we hope to use our sensors to tell me, is it the TB hypervariable region or one of these other species? So this is the visual version of that assay. So you can see by the visual color change that if you have sensors for abscessus, you only detect the abscessus DNA, but none of the other species. So we had very high level specificity and a much easier readout for a point of care setting that doesn't require any kind of fancy instruments, as long as you're not colorblind, I guess, because it is green. So the last thing that we were trying to do is to combine our sensors with amplification methods like PCR so that we could enhance the sensitivity of the assay. So without going into too much detail about the enzymes that go into this, we've explored a method called RPA, or recombinase polymerase amplification, that kind of like PCR, you have two primers, and then you have very rapid amplification of your target sequence. But this all happens at a single temperature. So you just need an oven or a hot block, and in 15 or 20 minutes, you can have very high level amplification. So we would amplify, say, 16S ribosomal RNA, and then use our sensors to detect it. So this just shows we've optimized the primer conditions, we've optimized the kinetics, so we know we can get detection in less than 1 hour with amplification, and then detection with our sensors. And so now we're kind of moving this assay forward to test for drug resistance and for other things that we want to detect. The last version of this assay that we are playing around with is back to PCR. So we have designed asymmetric PCR, where you have more of one primer than the other, so that in the end you make lots of single-stranded DNA that our sensors are the best at detecting. So this just shows amplification of genes to identify TB or to identify drug resistance. And we mixed all these primers in one tube, and I was amazed when we first did it that I read about multiplex PCR, but I was sure if you mixed 10 or 12 primers in the same tube, it would be a very ugly mess, but you can actually amplify. I think we've amplified up to 15 different gene loci in the same tube. So we can amplify any gene that you want to detect for drug resistance. And without going into all the details of the data, we're working on multicolor sensors. So in one tube, we can detect up to at least 3 different things at the same time. So we could tell you yes or no TB, and if it's TB, does it have also a mutation in this gene for drug resistance? So trying to make the assay more complicated, but in the way that one tube can give more information. So in the clinic, you don't have to do 10 different tests side by side to get all of this information. And the last little piece of our puzzle comes about because of Dr. Brian Kim's engineering genius. And so these are just some sort of images of a 3D printed real-time PCR machine that he's designed and built so that we can have up to 3 different fluorescent colors. And you have this little turret that will turn around, and so the tubes can be hit with different LED light sources, and you have the filters and detection units. And this whole unit is maybe the size of a large mate cup or something like that. It can fit in your pocket. It runs with a battery. And so as far as point of care implementation, that is one of our goals. This is an image of the very tiny thermal cycler unit that he's built that fits inside this machine, that is designed for very rapid cycling, so you can have very fast amplification. And this is just a picture of some early data of showing that we can conduct these assays in this machine. And a negative sample, you have a low signal. If you have a positive sample, you see this almost immediate increase in signal fluorescence. So that is kind of where we are going with these assays, of trying to determine the best combination of amplification method and detection method, and then combine that with a portable, user-friendly sort of device. And as I mentioned, this was in collaboration with folks down at PrInt-FURG, and the pandemic has obviously changed some of our plans of exchanging of people and bringing students here. So we've been doing, and even ordering supplies has been very difficult. So we've been doing the best we can to optimize things here, ship them to Brazil, and then the students there, now that they're allowed back to the lab, we're into a much better place where the assay platform is working. This is some data that was sent from the group down there with Ana Julia and Ivy and Marcos, and I don't know exactly who did this particular replicate, but so we're really excited. We have another 2.5 years on this project at least to hopefully advance this platform and test in the real world on sort of preclinical evaluation on TB clinical samples. And eventually, when we're allowed to travel again, I'd love to bring some of you up here and vice versa so that we can accelerate this collaboration. So with that, I will stop and be happy to answer any questions either about the science or about other collaboration opportunities. We do lots of stuff related to the ocean in Florida other than what I do, so I can connect you with a bunch of other people. Thank you very much.
Thank you, Kyle. I will leave to Andrea to coordinate the questions and the dynamics of questioning and answering. Thank you very much for your impressive talk. And I will let Andrea to proceed. Thank you, Kyle.
Okay. Thank you, Kyle, for the nice presentation. And when I look this presentation previously in another event, I look and say, oh, this is, you can combine several topics of our internationalization project. And for me, it's nice because there is the marine products, there is the development of diagnosed methods for a disease that also is very common in the coast, because tuberculosis also can be transmitted. And also you have some situation that here you have a harbor, a marine harbor that you can have the introduction of the tuberculosis here, and also to have other disease that allow that tuberculosis, that patient develop tuberculosis like HIV. So it's so nice, your presentation. And now it's open for someone want to do a question. So we have also the chemical products. You have a different topic here that the audience, you have different audience here that are able to make questions. And so if someone wants to make a question or a comments, so Pedro, you raise your, please.
Thank you, Kyle, for your amazing presentation. I have a couple of questions. The first, it is you have results in vivo of experiment, the heat, natural heat?
Not yet. That is sort of the next phase of things that at this point it needs some improvement on the selectivity. So some of these new analogs we think may be good enough for sort of early stage mouse studies. And so we were sort of gearing up to that. But we had a few more studies to do first in checking the tolerability in mice. So those studies require quite a lot of compound. And so now that we have some analogs that improve the activity on replicating and dormant, we think those will be better because they will hopefully kill all of the different phases of TB, but we haven't yet. We have a mouse model here that we can do that, but we just weren't quite ready yet with trying to make sure we pick the right, because it is a lot of work and it's not cheap to make sure we find the best compound. So we have some studies to look at sort of the microsomal stability to try to make a prediction. Will this be okay in vivo, for stability? Because if it gets degraded or metabolized too quickly in the host, it will be not useful. So I think our first attempt will be probably direct delivery to the lung, because then we don't have to optimize the pharmacokinetics for oral to the blood. We can do a test of in vivo efficacy by delivering directly through the nose to the lung, as it takes less drug. The toxicity problems are less because you're not giving systemically. And so that is typically our plan for these very early drug candidates. Is, it's difficult to get the good pharmacokinetics without a lot of work. So we can at least sort of get proof of efficacy in vivo by just putting it down the mouse's nose.
Okay, thank you. I have another question about this model of dormant, like real dormant environment.
Right.
Did you make with 9 days with this dormant condition? It's okay.
Right.
More 2 days with log growth, the logarithmic growth.
Say that one more time. Could you clarify that? Do you mean, was the assay we exposed?
Yes, the assay.
We assayed exposed to the stress for 9 days first, and then we have to remove from the hypoxia quickly, and then we add the drugs and put back into hypoxia for 2 more days, and and then you can see the loss of signal. And we showed back when we initially had, when I mentioned Carolina's like 1.5 year adventure, we were using first a fluorescent bacteria and doing the same kind of model. But we found when we thought we had a lot of hits against dormant bacteria, when we tested directly and plated for CFU. So you adapt for 9 days, we treat for, but then we plate directly. We found that many of them were false positive hits. So because in dormant, they don't grow, right? So with the fluorescent, we couldn't tell the difference between dead or alive. So we had to remove the stress. So we added buffered media, we put them in oxygen, and we would say, if you're alive, you should start to grow. And if you're dead, you of course stay dead. So then after 4 or 5 more days, we would take the signal. But when we tested those directly with no regrowth phase, we found many of our hits don't actually kill dormant bacteria. They were just very potent compounds that when you wake the bacteria back up again by removing stress, then they kill them. So when we switched to the luciferase, we could eliminate this extra 5 days of trying to regrow in happy conditions and directly see loss of signal. And so now we have very good correlation between real killing of dormant bacteria with the signal loss. So yeah, these assays are a little bit complicated because normally we would start the assay very low inoculum and you wait 5 days, and if you're dead, the signal stays low. And if they grow in 5 days, you can have a big signal. But with dormancy, they don't grow.
Yeah.
But now with the luciferase, because it depends on ATP and protein production, when they're dead, we can see directly the loss of the luminescence. So yeah, it's just a model. We know it is not perfect mimic of the lungs, but
in fact, the last question, I have a concern about the selection of drug-resistant strains in the treatment only one drug. It is. I think that it's a big problem.
You mean sort of the in vivo situation where you're not actually hitting with 4 in the same place?
Yes, I think that it is the treatment of just isoniazida, for example. It is very important to make the treatment with the TB in latent stage,
Right.
not active TB, because it's the population of heterogeneous population of strains can be its reason for drug-resistant strain selection.
Right. And so, yeah, I think that having a combination of drugs that can kill these dormant and the active replicating, because they're both present, and finding the right synergy combinations to, best kill so many different TB. And people will test against TB and macrophages because that's another model of in vivo environment. So that's one of our sort of assays at a later stage, that we will try to make sure that these compounds can go into the macrophage, and whatever the bugs are like inside a macrophage can also kill them, because they change efflux pumps in different circumstances. You never know. So we like to test them in multiple models and try to prioritize the ones that kill everywhere. But
okay, thank you.
We have some
question in the chat.
And the chat, and also
I see one question in the chat from Daniela about the challenges of working with natural products. Yeah, this was an area that this first project ever for me with drugs and TB was the marine natural products. And we've had several different collaborators and invested quite a bit there. And I think it's it's obviously the source of many of our existing compounds. Nature makes compounds that chemists struggle to make. So I think for finding new scaffolds that are biologically active, I think it's a great place to start. But yes, we have already sort of run into the problem of scaling up is a problem. So that is the biggest downside for the, source is the bottom of the ocean with a very limited resource that is hard to go get more of. So that from the marine environment, if it's from a large organism, not a bacteria from the ocean that maybe you can culture in the lab, that is one of the first hurdles that we and anybody would encounter. Those that you can culture, like the fungal sources or bacterial, I think sort of minimize that problem. Then it becomes you can scale up and produce more of that one compound, but to advance any natural product, you're very unlikely to take the natural product and go directly to humans. It will need lots of modifications to improve the pharmacological properties. So then you have to find a brave chemist who wants to make it, synthetically in a way that is, scalable, that you can make analogs. And so I think that can be difficult. And so the best way I have thought to do that is have the best assays at the beginning, so you have the most data to prioritize your effort, and have multiple choices to send to the chemist, so they can, maybe there's one or two that they say, I think I can make that. Then the chemist can, from a synthetic perspective, say this has potential to, like with this molecule. Now at least we have some way to go forward. Otherwise, many natural product studies stop at a very sort of early stage, because you can't make analogs and you can't move to the animal model. And so for me, that's been the challenge of the natural products and there are chemists out there that like the challenge. And so you just have to find them and find somebody willing to work with you and to make these products. But
and Ines make a comment in the chat, but also Ines, if you want to make a question, can please.
Yes. Can you hear me?
Yes.
Okay. Thank you, first of all, very much for this very interesting talk. And I'm coming from a different direction of research, but automatically caught my interest when you talked about dormancy in bacteria cells, because I'm working with senescence cells, no? Cellular senescence, especially stress-induced. That's what you do also, no? You stress your bacteria to induce dormancy, no? So therefore, I saw a lot of parallel things, but I'm not sure how much connected, how much similar or different these processes are. So my question was, you said you are interested in other drugs to test this, maybe accompanying the treatment of TB for the active cells and to have a accompanying drug to attack the dormant cells, no? And the senolytic drugs, I don't know how much you know about them. Some famous ones are quercetin, dasatinib. We know them already in mice models. We use them already, so you would not have this test phase.
Right.
And they use them because senolytic drugs, they somehow put the cell back to the apoptotic pathway. But I don't know how much different is bacterial dormancy to cellular senescence.
Right. Just because of how strange TB is in general, even compared to other bacteria, I don't know the details of the senolytic, pathways or mechanisms. I would guess that are no molecular parallels just because of the divergence of the genetics of TB, and it does have a very elaborate, it seems like, program involved. And so we at least know experimentally that if you throw TB in hypoxia very fast, it will die. But if you give it time to change and adapt, then it can live for a very long time. And people have studied all these genetic programs that happen. I don't anticipate there would be direct parallels, but the idea of, can we find a way to sort of force the TB to wake up so that you can reactivate them and kill them, was something that somebody brought up at a TB conference a few years ago, that they had this idea, but were sure nobody would ever fund a project to try to wake up dormant TB, because that could be dangerous unless you're very sure you can kill them once you wake them up. And so I think there are people thinking about those concepts, but so far at least we don't know how to, like with a small molecule, force them to, become active again, because there's so many stresses telling them to stop growing and to stay quiet and just hang out until, the conditions are better. I think it's an idea that people have explored, but I haven't seen anybody actually work on that yet.
Yes, thank you for your answer. Thanks.
The only one I know, there are some factors called resuscitation promoting factors in TB that are involved in cell wall maintenance and remodeling, that people would say that if you put those on bacteria in certain models that are not replicating, that they will trigger resuscitation. So that's the closest thing I've seen in our field, is there are some mechanisms that may be involved in like restarting growth, that maybe in the future, when we know more basic biology about the bacteria. The other, maybe not direct, but there are a lot of people trying to go for host-directed therapies to program the cell to better kill the bacteria. So to drive autophagy or something like that as a way to use the host against the bacteria. But it's a little bit different than I think the senescent idea, but thank you, interesting.
Thank you so much. There is someone that wants to do a comment or make any question. So.
I put them all to sleep.
No. So I think that we can finish our session. So thanks to everyone that participated of this presentation. And so.
Yeah, thank you especially to Kyle for accepting our invitation to give this talk.
Thank you very much.
And I hope our collaboration will increase in strength.
Hopefully so, yes.
Thank you. Have a good day.
Thanks. Well, I think that you can finish the session, and now you can come back in the afternoon.
Yes, we resume at 1:30, right, Andrea? With
Yes.
Martin's talk.
Martin's talk, and this room it's open all the time, so I'm not close the room. So if people want to keep open here, so we can meet at 1:30. So
Thank you. Thank you, Andrea, for conducting the session.
thank you to everyone.
See you later.
See you later.
See you.
See you.
Congrats. By the morning.
Okay. See you.
Andréia, você ainda está por aí? Edu?